Thank you so much, Professor Shantanu Chaudhary and Professor Mitali Mukherjee, and of course, my friend Professor Manoharan. I just wanted to, congratulate the speakers who spoke before me. I mean, what a galaxy of stars! Some of my heroes from the scientific world: Professor Partha Majumdar, Professor Rao, Professor Brahmachari, Professor Ashutosh Sharma. I've looked up to their work and their leadership in taking forward uh, the whole agenda of science and scientific thinking. So I'm actually pretty much in trepidation and trembling just now because I don't know. It's a tough act to follow. Um, but let me try telling you about what I think about a story that has been plaguing us for the last uh, more than 2 years, perhaps, and that's the rise of a virus that has unsettled the world. But I'm going to take a position from the other contrarian view about the microbial world and, in particular, viruses. Um, this talk is based on a book that I've written, and the book itself is based on a podcast I did in May 2020. So I just want to take you first to an image from a French encyclopedia for housewives. And this image is from 1880. What this image shows is that there are uh, three children with three different types of clinical manifestations of the skin. The one on the left is measles, the one in the center is scarlet fever, and the one on the right is smallpox. So the reason why I bring this up is this that soon after this phase of literature or writing, the the focus of medical research and writing shifted because we entered an age of discovery, and not only the discovery of what microbes were or even viruses, but also the age of antibiotics. And within 30 years of discovering effective antibiotics, it was summarily dismissed in most medical textbooks. For example, in the 1962 textbook of infectious disease by Castle, it says that the end of the infectious diseases are near. And as a result, there has been very little uh, study or emphasis on many infectious diseases that have been persistent or those that have been emerging. Um, this image in particular becomes very, very important for us now because it's a reminder that we may have pushed ourselves back into a more primitive mindset when we start exploring the fact that we have encountered pandemics. Um, like I said, pandemics were considered, or even outbreaks and epidemics, at least in more developed parts of the world, were considered to be a thing of the past. The emergence of COVID-19 has shown us that we may have not passed that threshold where we can discount the role of nature and, more importantly, the role of the microbial world, whether it's in a positive light or the detrimental uh, things that it can throw at us. So I'm just going to say that while we look at the negativities of the microbial world, we changed our narrative drastically. From just a year prior to the outbreak of COVID 19, we were celebrating the discovery, or rather the announcement that a major uh, uh, progress has been made in terms of um, new microbiological techniques like using of CRISPR, and for which uh, the Nobel Prize for Medicine was given. So it's been a bit of a volte-face in terms of how the world has viewed at one moment as being euphoric, and at another moment at this being a time when we have to see ourselves in the perspective that the microbial world has reset for us. I'm just going to be very quickly trying to tell you what I mean by the microbial world. If you were to look at under a microscope and take a pinch of a moist uh, uh, soil from, say, a pond or a bird bath, chances are that you would encounter a worm like this, which is called a nematode. Now, nematodes are really, really tiny. If you have to really gaze with your naked eye to try spotting it, but it's rather simple to see it under a microscope. It's very elegant. It's the most abundant macroscopic creature or in the world, in the sense that this is the most abundant metazoan uh, in the world. It's reported that about 57 billion nematodes exist for every human being. Um, this worm-like creature is always pretty simple to find in pond water or on the edge of soils that are near ponds or bird baths or even rivers. Up next, if you were to bring down your field of vision a little more by, say, 10 times more, you encounter these slipper-shaped creatures. These are parameciums. I'm sure most of us who've studied biology would remember this creature. They slink around over one another, and they are extremely mesmerizing to look at. If you were to move your field vision a little along the slide or uh, the drop of water that you have prepared as a hanging drop preparation, chances are that you might encounter very beautiful geometric creatures like this one. Now, this is a solitary living algal body called Microasterias. Now, Microasterias means micro means small and asteria means sun. So it looks like the sun, and it's under refracted light. It's absolutely beautiful to look at, and it can come in various colors. It can be greens and yellows and reds, and so the pigments depend on the day, time of the day when you're looking at it, and also the species. Uh, if you were to uh, narrow down your field of vision, and in fact increase the resolution of your microscope, you would encounter some fuzzy things like the one that is on the foreground of this image. Now, this is a stained image. None of these images are true or taken in one single vision. Let me put that righter. These are collected and seen under different kinds of uh, microscopes. So it's just a collection of images. Please do not treat them as having been seen in one single vision. This is a stained slide, and the creature that you see in the foreground with the white, the black and gray strands with what looks like heads of flowers is actually aspergillus. It's a deadly fungus in humans. Some species, 3 species, can be deadly to humans, but the other 80 odd species have no effect on humans or are actually soil dwelling fungi. The red or the pink colored single cell that you see is a yeast cell, Saccharomyces. Now, Saccharomyces can live in groups, in bunches, or they can be solitary. But what is critical to us is now looking at the smallest creature, and this is uh, the virus. Uh, these are multiple viruses trying to attach themselves, and they are in a race to send their genes through the cell wall of this bacteria. There's a multitude of viruses, a variety of viruses, but most cells and most creatures have very specific viruses, very specific to their individual cells and organs and even body types. So, having seen the diversity of what you see under a single drop of water, I just want to reiterate the fact that this about the size of different microscopic things that we encounter in our daily lives. To your right is a strand of hair. I mean, just to show that it's an average human hair, which could be 50 to 180 micrometers. But if you were to move towards the left, you come to the two disc-shaped red-colored objects that you see, and that's the red blood cell. Let's move along further to the left. You see these two blue sausage-shaped uh, uh, bodies, and that's average bacterium. But let's move a little more left, and you see this brown spherical object, and that's wood smoke, the smallest particle, one of the smallest particles that the human eye can see. But again, let's uh, move a little more left, and you see that red dot that's on the left, and that's the coronavirus. It's one of the larger viruses. I mean, it's not the largest, but it's one of the larger viruses that we know. And to the left of that is the bacteriophage, the one that I showed you previously in the slide where the bacteria was being attacked by viruses. Such viruses are called bacteriophage. They look like space landers. I have an image later which I will show you. And to the left of that, it's barely visible, is a Zika virus. Zika viruses are among the smallest known viruses that we know of. Of course, polio is also an extremely small virus. Uh, but while viruses are really small, we also have giant viruses. I want to take you to a small story that started in 1992 on the top of a cooling tower above a hospital in Bradford in Yorkshire. Now, Bradford is an industrial town, and many of you would know Bradford because of Jack the Ripper. There's very little else that is memorable about the town. But for virologists, the discovery that happened here, this was incredible. Now, in 1992, there was a mysterious pneumonia that was happening to people who had, or patients who were admitted in the hospital, who were slightly low on immunity or were immune suppressed. And this pneumonia, the cause of the pneumonia was remained undiagnosed for a very long time. It wasn't lethal, but the pneumonia was persistent, and the normal antibiotics that were being administered did not seem to work. The pathologist who was entrusted to find out what the cause of the outbreak was found that the creature the life form that was isolated from the lungs was also found in the cooling towers of the hospital. But no matter how much he tried, he was unable to culture it. That means he was not able to grow it in the nutrient agars and the medium, the different microbiological mediums. The standard practice is that if you are able to grow it and you can see it under the microscope after a process called staining, what is called gram staining. So he nevertheless concentrated the cells that he was able to isolate from the lung samples and tried to stain this. And he found something very peculiar, because staining process generally gives a single color, either blue or pink. But in this case, he got both the colors in the cell. So clearly, this was a very different bacteria, if at all it was a bacteria. Uh, he looked further and tried using other tests, microbiological tests, but he found that this was something very different, and uh, unable to diagnose it any further. And given that there were huge cuts in the NHS, the UK government in 1993-95 had major fund restrictions, and because of which the scientist sent a sample to two French scientists in University of Marseille, and there it was languishing for about a year or two. And it was only after somebody remembered that there was this mysterious fever outbreak that happened in Bradford. Let's look at that sample. And lo and behold, they found the same thing that they were supposed to not see. These two pink and blue cells. They decided to put this under a electron microscope, and then they discovered something like this. They found that there were giant viruses in the center of these rounded cells. When they teased out these viruses out and let the circular, uh, the white matter in which they were living, they found that the host was actually an amoeba. It was something called an acanthoamoeba, one of the most primitive forms of amoeba that is alive today. Um, and once they published this in a paper in Science, uh, there were other discoveries that were reported from a thermal spring in Japan and even from IIT Bombay from Powai Lake. You know, and since then we've got about 20 odd species of these giant viruses that have been discovered. Now, these are rather simple to see even under a simple microscope. So the giant viruses actually can be pretty large. I mean, they can be giants, right? I mean, in the microbial sense. What makes giant viruses unique or really relevant for us is that they have turned around the conversation on how and when and what would have caused the evolution of viruses. I'm not going to go into it, but I mean, I'll do some same shameless promotion of my book. I mean, if you really are interested in how viruses evolved, uh, did the cells evolve first or viruses? It's like a chicken and egg conversation, but you'll have to read my book. So, um, so what we now know, because of the nature of which we've studied the structure of viruses and the genetic structures of viruses, we now know that there is a variety of shapes and even sizes, and also the types of genomic structures they have. So it can be DNA or RNA, they can be double stranded or single stranded. And even then, there's complexity. There's something called positively sensed and negatively sensed. With so much diversity in the microbial and, in particular, the viral world, the classification body of virologists, which is called the ICTV, International Committee on Taxonomy of Viruses, in 2020 decided to expand the seven-tiered Linnean classification model to 15 uh, rank and model for viruses. And while this would upset the lumpers, but there is no other way to accommodate the complexities that the viral world shows to us. I just want to make 5 propositions to you. My first proposition is that the viral world is an integral part of the microbial world at large. And they provide services that are absolutely unique and fascinating. And if we were to put them in the right perspective, in how they manage to balance the various forces of nature, we would have a greater appreciation of it. And my first contention is that the very breath that you and I take is made possible because of microbes, and the role of viruses in particular is incredible. Let me show you this image. Some of you who've read my book, my previous book, Indica, would remember this image. Now, this is the reclining Vishnu from Bandhavgarh. And Vishnu, as you know, is the god or the preserver, and the god who maintains the balance in nature. Now, this statue is overlooking a placid pool. The pool is covered with what you and I would call slime, but for a microbiologist or somebody who studies algae, this is blue-green bacteria. Now, blue-green bacteria are incredible creatures. There are at least 8,000 known species of blue-green bacteria, and they dominate the top 8 meters or more of ocean and sea water. And in lakes and ponds in fresh water, it can be the entire horizon that can be dominated by these bacteria. What is incredible about these creatures is that they evolved about 3.7 billion years ago, and they were found in the margins of wherever water met land. And they colonized and partnered with other bacteria especially specialist bacteria that could break down and assimilate specific material and nutrients or elements. This a, is a fossil of a partnership. Now, this kind of a rock which entombs the fossils of cyanobacteria from 3 billion or older years is called a stromatolite. Now, stromatolites basically are layered rocks which show that layer upon layer, cyanobacteria and a partner bacterium was collaborating to grow into colonies. And a single rock like this, which is about a foot wide and long, is about, I mean, it covers at least 10,000 to 15,000 years of uh, constant growing. Now, what was remarkable about stromatolites was that they produce so much oxygen for the first 2.5 billion years that they created Earth's atmosphere. They also caused the oxidation of most metals, for example, iron oxides and calcium oxides and all those oxides that you see. Prior to this, most elements and most minerals were very simple and basic. There were no oxides. And the third thing they did was they created a ozone layer. They produced so much oxygen during the 2 billion years that they dominated. They created the first major uh, ice age of the Earth. Now that was the key uh, event in Earth's history to not only produce all the water that we have in oceans that are filling the basins and rivers that we have in the world today, but it also created the the chance or the opportunity of uh, leapfrogging from unicellular life to multicellular life. Because once that availability of oxygen and oxides and the ozone layer protecting vulnerable cells from expanding and accreting together to create more complex life, uh, this whole process enabled the evolutionary leap for more complex life, especially multicellularity. Now, having done this, all this oxygen being made available and triggering more complex life to arise, we find that the blue-green bacteria continue to dominate the ocean waters. Now, as human beings, we tend to focus more on land. We love to see our forests and our deserts and our grasslands, but very few of us actually pay attention to what happens in the oceans. Most of the oxygen production that happens, most of the free oxygen that is produced also, is not from forests or grasslands, but from the oceans. You remember that I said that 8 meters of seas and oceans are actually dominated by single-celled creatures. And among them, there is a creature that you see along the equatorial belt, the bright green colored swathe that you see across the globe, is, is a champion organism called Prochlorococcus. It wasn't discovered till the mid 1990s, and this is the most abundant cell outside of the viruses. The Prochlorococcus and its cousin Synechococcus, which you see in sky blue color, is perhaps the two organisms that work together and produce anything between 20 to 24% of all the oxygen that is produced on Earth. Now, what is remarkable about the blue green bacteria, the varieties that exist, is that they have a very short lifespan. And because they are in the ocean and once death occurs, their cells sink into the depths of the ocean. Now, let me digress a little and tell you that our fossil fuels that were made, 300 billion years or, 300 million years to about 200 million years ago, um, are all embedded in deep into the earth. For example, oil and gas and coal, all of this lies in the depths of the earth. Once they are taken out from there and burnt, they're consumed to produce energy. The carbon dioxide stays inside the atmosphere. That's the cause of concern that we have, and that's the reason why we have global warming and therefore climate change. The mitigating measures that we have proposed is to plant trees and have direct to air capture. The challenge is that both these measures would leave the carbon on the surface, right? Because trees uh, accumulate carbon if efficiently, but they also stay on the surface and they are prone to decomposition and worse, to wildfire. Now, in either situation, the carbon gets released very quickly. As we speak, there's a massive fire in Argentina. And last year, we had massive fires in California, Australia, Russia, and even for the first time in the permafrost region of the Arctic. Now, with so much carbon dioxide exposed, whether it's organic carbon dioxide, carbon in the form of vegetation, or it's in the form of other things like fossil fuels, the burning makes sure that the carbon dioxide gets released quickly. And for this reason, organisms that are on the top of ocean layers and lakes and ponds and go and bury themselves after they die into the depths of the oceans or lakes stay there for thousands and perhaps even millions of years. Some of them even get converted into petroleum. So the efficiency by which carbon gets buried is in the seas, in the high seas. And therefore, the role of ocean, seas, rivers becomes even more important if we are looking at mitigation. Now, where does virus fit in into this story? Now, this is, I told you that the champion creatures are Prochlorococcus and Synechococcus, right? Now, these, like I said, anything between 20 to 24% of all free oxygen is produced by them. But their life cycle in normal circumstances would be 2 to 7 days. But the presence of viruses like these, they have very specific viruses, and for each strain of Prochlorococcus, there is a specific phage. Remember, I used the word phage as in the person, as in the virus that eats a bacteria, bacteriophage. Now, bacteriophage accelerate the killing of bacteria, right? Now, Prochlorococcus and Synechococcus have very specific viruses that kill them. And so is it for some another creature called Trichodesmium. Now, Trichodesmium is in the oceans the most important or the most efficient organism that digests and assimilates and recycles nitrogen. And Microcystis does the same for sulfur. And there are several other uh, microbes and their viruses that ensure and regulate this process. So the viruses are efficient undertakers of this whole process. The whole ocean life cycle, and also on the surface of lakes and rivers, and also moist soils, these creatures are working together, one predating on the other, but ensuring that there is a constant balance in their numbers, which keeps the oxygen carbon cycle in balance. And the same is true for Trichodesmium, which does it for nitrogen. The same is true for Microcystis, which tries to manage or regulate the entire sulfur cycle. So, the beauty of this finely calibrated, interlinked cycle of elements, nutrients, and minerals is orchestrated between the viruses and different microbes. And if the viruses were to go on a strike, say they decide to go for a strike for a year, or forget a year, even a month, there would be a massive oxygen crisis, and our oceans and seas would start looking like the sewage lines that we have in our country, right? Thick and piling up with green gunky material, right? And that is what's going to be the fate of water bodies. So we must accord some responsibility and some importance to this delicate relationship between the viruses and the larger microbial world. So I hope I have made some um, uh, my first proposition of trying to convince you that the free air that you and I breathe, the free oxygen that you and I get with each of our breath, is courtesy the processes that are happening in the open seas, and that's how the oxygen and the carbon dioxide and the free uh, uh, composition that we have is largely because of how these creatures orchestrate their processes on a daily basis. I'm now going to move to my second proposition. I'm going to make a very, very strong claim. My submission to you is that after the sun and plate tectonics, it's the viruses and the microbial world in general that have powered evolution forward. Other than the oxygen contribution that was done by the stromatolites and the blue green bacteria and the viruses, there is something even larger that has been done in the past. And I'm going to start with a story of this fish on the left. Now, this is a coelacanth. It was discovered accidentally. It's in a deep sea trawling event that was happening in Comoros Island, and it was a chance discovery that was made by a family of South Africans called the Latimer family. Um, Now, coelacanth is a very unique fish. It possibly emerged around 410 million years ago, well before the time of the dinosaurs. Now, it's a fish that lives in submarine volcanic regions, and it lives in caves. You can see that it's in a hollow-like structure inside in deep sea. It has very unique scales. It has very unique eyes. What is really unique about it? It's got a muscular fin and a very muscular tail. Now, deep sea divers who have photographed and pictured the movement of coelacanth have narrated that they've seen this fish actually climb the vertical walls of caves, trying to find small hagworms or even small plant material for its daily consumption. Uh, the muscular fin and the muscular tail is something that is unique to this fish. And there's only a couple of other fish, like the lungfish, that have this. Now, so what is really important about developing a muscular fin is that a chance infection that happened to an ancestor or a predecessor of the coelacanth caused this fin, which is otherwise non-muscular in other fish, became muscular. Now, the infection that happened was the one that you see on the right. Now, that's the virus that caused it. Now, it's called a foamy virus. Now, foamy viruses are RNA viruses. So once they infect, they cause the tissue to become a bit bubbly or like foam, and that's why they're called foamy viruses. And in some instances, the RNA of the virus goes and integrates in the DNA. And that's when this incredible leap in evolution happens. A muscular fin and a muscular tail were the essential ingredients for the first steps to take place on land. So the amphibians also have a foamy virus. Amphibian, the ancestors, or rather the descendants of this fish, or something like this, would have created or or would have evolved into something that became an ancestor of the amphibians, carrying this foamy virus and then adding new foamy viruses to develop a forelimb and a hind limb and a muscular tail. And that creature, or a creature something like of that nature, was the one that eventually took the first bold steps out of the sea and colonized land. And since then, there have been subsequent and repeated foamy virus infections, causing the physiology of all creatures to constantly evolve and create new kinds of limbs and musculature and symmetries. Now, this is only one aspect of that infection. I want you to bring an infection that happened, which is which you might relate to more closely to. Now, this is an image from about 255 million years ago. This is the time when the land was dominated by this creature on the left, which is called a Lystrosaurus. Now, this is an amphibian about the size of like your auto rickshaw, right? And these were grazers. They needed to come close to water to lay their eggs and for their young to grow. But they moved in herds, quite like our cows or antelopes, and they would graze on cycads and ferns and lycopods. The creature on the top right is the ancestor of the modern day alligator or crocodile. But the creature that we are interested at the moment is the one at the bottom right, one which is standing on top of another lizard like thing. And it looks a bit like a mongoose, but this is a true reptile. But there are signs to this that show that it is not really a true reptile like the one we imagine. It does have a scaly mouth and it has hollow fang-like teeth. It has a scaly tail and scaly legs, but it also has hair-like protrusion on its back. Now, this is the first sign that you see in this creature called Thrinaxodon. We found fossils of Thrinaxodon in Andhra Pradesh, for example, and even in Jharkhand and Chhattisgarh. Now, this is where we find the first signs of mammalian signs of developing uh, uh, hair on a scaly skin. Now, why is it important that we're talking about this? Now, mammal-like reptiles diverged into two broad paths. The first stayed very reptilian, and they became the ancestors to the monotremes. Now, the monotremes, like the platypus or the echidna, still lay eggs, and they have a poison gland, and they have spurs on their hind legs. They also have a scaly underbelly. So they have a lot of similarities with the reptilian ancestor that you see before you. The other stream that evolved is the true mammals, that is, like us, or the dogs, or the cats, and the camels, and all of those. And they developed what is unique to uh, true mammals, which is a placenta. That means the ability to give life birth. Now, I'm not going to go into the history of the placenta, but placenta is a fascinating organ. It is completely developed by a protein that is of viral origin. If that viral protein is inactive or does not or is absent in a person, which is highly unlikely, the chances of that female to conceive is next to nothing. There is also another uh, protein, a viral protein embedded a viral gene that is embedded in our genome, which protects us from uh, causing spontaneous abortions. Now, remember that this is a viral protein that is creating an entire organ using nutrients from the human body, right? So everything that it does inside the body is foreign. And the chances that the human immune system rejects it is very high. But in order to keep the placenta intact, there is another viral gene that actually modulates it down, your immune system, from overreacting and ejecting a placenta. So, so in a sense, it's paradoxical that our own birth is something that is concocted or orchestrated or conspired by the act of viruses. So it feels like the film Alien, right? Where you have alien injecting something into your body and then it multiplies inside you. Well, that's exactly what happens when a person conceives. This protein gets triggered, and a placenta is is formed. I'm now going to present to you my third submission, which is on defensing. Defenses that are created by viruses themselves. Now, viruses are known to us as pathogens, but there are more viruses that do more good than viruses that do bad to us. And this is a very, very interesting area of exploration in science and something that is not often talked about. Now, as soon as we are born, if we are born through a normal birth canal, we get laced with lactobacilli and several other uh, important microbes that we get from our mother. But even as we are fed by a mother's milk and also by diet that infants are given, we acquire a microbiome which is very similar to our own parents. Now, that microbiome also has a very rich signature of viruses. I'm going to first show you this image. Now, this is herpes virus. We know of eight herpes viruses that affect the human populations, and we actually have anything between 2 to 6 of them at any time. Chances of us getting, say, chicken pox or herpes zoster, which is a very painful condition. Herpes zoster is basically known as shingles. It happens when you're older and slightly in your 30s, 40s, or 50s, and you get this on one side of your body. Now, this is a virus that actually lives in nerve ends or at the end of your cutaneous and subcutaneous boundary. What it also can do is start circulating inside your bloodstream. And there it has a role that until 2016 was not known to virologists. What we now know is that a rich viral load of what we now consider are benign viruses, which otherwise do not cause disease, are actually uh, beneficial to a host because they keep the immune system primed, but they also lock out viruses from entering you. For example, if a receptor has been occupied by a virus that is circulating within your body, chances of another virus utilizing that as a portal of entry is therefore negligible. Now, what we also know through several genetic studies of viruses, but also increasingly the DNA that Dr. Partha Majumdar was talking about, uh, such bidirectional studies of genomes of viruses and humans is informing us that there have been multiple exchange of both disease-causing viruses as well as beneficial microbes across our human ancestors. And this 7 million-year-old uh, family portrait, if I can say that word, from our earliest ancestors who began to walk upright uh, to the modern day ancestors. And as Professor Partha Majumdar said, there were at least four known um, uh, uh, cousins of the Homo sapiens that lived alongside them, and which enabled a constant exchange of microbes, especially of viruses. And that enrichment has been something which is important because it has not only made us susceptible to disease, that's the popular theory that we follow, but we do not look at the underside, that there is a lot of benefits that the other viruses that we are now discovering that have actually protected us from other more, uh, possibly more, potentially more uh, lethal infectious diseases. And among them is this virus. Now, it doesn't look like much, right? It looks like a cluster of just uh, gray blobs. And this is called anilovirus. Aniloviruses have been found to be extremely beneficent in people who have low immunity or are immune compromised. And people who have a high titer of aniloviruses have been found to be protected from otherwise uh, extremely lethal infections that they would have acquired in the hospital or during the operations that they have to undergo several times. So, uh, carriers of anniloviruses, of different varieties of anniloviruses. Now there are about 40 known anniloviruses in humans, which are found in different people. So this is an area of immense importance. What was once thought to be extremely sterile, for example, the cerebrospinal fluid or even the human urine, we now know that there are several varieties of viruses that actually inhabit our bodies. And it's only a matter of time when we actually start cataloging and discovering the benefits of viruses that live within us. I'm now going to come to my fourth submission, which is on order and disorder, the balance of how viruses and the microbial world survives within each individual. Now, until not too far ago, we used to believe that there were 100 trillion bacteria that lived in our body, as against 10 trillion human cells. Now, that was a guesstimate made in a correspondence in a journal, which actually found its way into medical textbooks, and then it became a bit of an urban legend. And that was a number that everybody who studies the microbiology or virology uh, tended to believe. But then in 2016, scientists in the Weizmann Institute in Israel decided to test this. And they found that there are 39 trillion bacteria to 30, as against 30 trillion human cells. That means for every human cell, there is 1.3 bacterial cell in our body. That means 30% more bacteria live in our body than our own cells, right? But in terms of mass, it is really tiny, because we've got cells like the fatty cells. They are very large. They can be few centimeters or few inches long, fatty cells, or even muscle cells and nerve cells. They are very large cells and they're bulky cells. And as a result, while the volume of our own cells is large and the mass is large, the numbers of bacteria are much higher in a human being. So 39 trillion bacteria to 30 trillion human cells. I mean, that's the ratio that you must remember. Now, what we now know is that the dominant, if you were to ask a dentist, I mean, try it on your dentist sometimes. If your dentist has studied in the 60s, 70s, 80s, or even in the 1990s, ask him or her, what is the dominant bacteria in our mouth? Chances are they will say it's Pedococcus. Well, we now know this is a cross section on the human tongue, right? This is actually a top view of the human tongue. And the different colors that you see are different colonies of bacteria, and they reside there and they perform specific functions. For example, if you've had something really hot or something really cold, if you had something very sour or bitter, different bacteria will act up and ensure that it does not uh, damage your tongue, or more importantly, start neutralizing what you've consumed before it reaches your stomach. So the whole process of digestion and pre-digestion begins with your tongue, and these organisms that you're seeing, this beautiful colored image, false color composite, is of a human tongue. And the immense variety of organisms are there for a specific purpose, right? So the discovery of the microbes that live within us is a very recent phenomenon. The multitudes we contain are unknown to us, and they decide about everything that we do. It decides on how much mass we are going to have. But just looking at the microbiome, scientists are able to decipher whether you are within the normal BMI or not, whether you have the Monday morning blues. That's the range of estimates that scientists can make if they have your microbiological profile of your different organs of your body. So it's getting sophisticated, and organisms, as they get discovered, are a true revelation of who we are. Let me just also reiterate that I studied microbiology in the 1990s, and we were told that the dominant gut organism is this creature called Escherichia coli. And this is the most studied bacteria in microbiology, at least human microbiology. Agrobacterium is for agricultural microbiology, and for human biology, it's Escherichia coli. It's a standard organism, that test organism that we use. But it's now been discovered that Escherichia coli is easy to culture, and therefore it was estimated to be the most dominant, the most prevalent organism in your gut. But if culturing procedures and ways to isolate and growing could be improved, and as they have happened, we've discovered that the dominant creatures are actually something else. It's Bacteroides. This is an organism that is important and seen most often in people who consume more meats and eggs and milk. And then there are two other organisms called Prevotella and Ruminococcus. Now, both of these are seen in people who take more fruits and vegetables and high fiber, and their proportions decide on how healthy your gut is and how good your systems are, and whether you're going to gain weight in the future or not. Of course, it depends on how much you exercise and many other factors. I'm trying to put it rather simplistically, but what I'm trying to drive at is that the organisms in your gut actually decide who you are truly. But for a very long time, it was not known that these organisms uh, exist, and how do they work together, and how do they colonize the different parts of the human gut. But the fundamental question that intrigued scientists was that if there is so much fermentation, digestion, assimilation that is happening in the stomach after we have a hearty meal, why is it that our stomachs don't burst open? Because after all, these organisms are all competing for food, and they produce gases and they produce acids and all those things that we know that these individual bacteria can do. Well, genetic studies in 2017 from across different countries that were analyzing for viruses in human normal feces or normal human subjects found regularly uh, traces of viral genes that showed that there was a single class of dominant virus, and it was not known for a very, very long time because it was very difficult to isolate it. But then they found, using computer sciences and genomics, uh, and they identified this virus, this class of viruses called the crass-phage virus. Now, it sounds crass. It's actually spelled as crass, but it's a CR small with A, capital A, and two small S's. So crass with a capital A in the center. Now, what does it stand for? It stands for computer assembled viruses. So CRASS. It's so computers were used to piece together the different genes that they isolated from human feces, and put this together and said we are looking for a virus that will have these genomes. And lo and behold, they searched and they found this in copious numbers. So think about it. The viral overload that controls our gut and the composition of the microbial residents in our stomach and our intestines and everywhere else was unknown till 2017, and we have given it a name which is also not something based on the features or the physical or the biochemical properties of the virus. We've given it a name which is largely on the way we discovered the virus, which is crassphage. I want to bring to your notice this bacteria. Now, this bacteria is called Ralstonia. Ralstonia was discovered in 1890 in Brazil, when a lot of food crops from South America were being transported all across the world, especially Africa and Florida and Southeast Asia. Now, this creature is something that was found in the soils of Brazil and Argentina and the Central American region, and it was known to infect eight root crops and some stem crops, but largely root crops, especially potato. Now, what is unique about Ralstonia is that it's an extremely mobile bacteria. It's uh, it's got a tuft of flagella at one end, which makes it extremely you know, motile. And as soon as it rains or there is a splatter of water, this bacteria is able to find its way to a new plant. It's extremely, extremely pernicious and ubiquitous. What we know now is that from those 8 plants that it infected, there are over 400 plants that it now infects in the New World. For example, tea. Now, there was no tea in South America, but the tea that it encountered in India, Sri Lanka, and China is now devastated by this bacterium. And it continues to infect potato globally. In fact, the damage that Ralstonia causes exceeds $5 billion every year. It was the first bacteria to be added to the US bioterror list. But as this bacteria moved into new countries, it also found a new predator. Now, this is one of those predators. Again, this is a bacteriophage. Now, bacteriophage, like I mentioned, look like space landers. They come and land on a bacteria. They are very specific. Unrelated bacteriophage will not infect this bacterium. And of course, it's a completely chance event because viruses do not have chemotaxis, at least we don't know of that. And it's a completely random process of how they will land on the cell surface of a bacterium or any other cell for that matter. So it is that chance event that has to take place for the bacteriophage to actually go ahead and infect Ralstonia or any other bacterium. But once it does that, the annihilation is complete. And they create several copies of themselves, which are then ready to infect more of the host that they infect. Now, this is something that has been of immense value to or it can be of immense value to the humankind. Now, why do I say that? Before the discovery of antimicrobials, antibiotics in particular, viruses, particularly the bacteriophages, were critical to fight infectious disease. Between World War I and World War II, bacteriophages were used in the trenches where soldiers were fighting, especially to fight against diarrhea, if they developed diarrhea, or if they had battle wound, especially gangrene. There were specific phages that were identified and they were administered, and uh, Polish soldiers, German soldiers, especially General Rommel's cache of arms and medical supplies that were found in Tobruk, found vials of bacteriophages, and it was clearly marked. And this is to be given in the case of diarrhea. This has to be given in the case of wounds. It was very, very specific. And there are only a few countries that continue to practice the phage therapy even today. Mainly Georgia, Poland, Russia, few other countries where phage therapy is legitimately used. Phage therapy is also legitimately used for Staphylococcus aureus in skin infections in cats and dogs in the US, but it's not permitted for use in human beings. Now that's a travesty because you're not using nature to fight nature, the problems that can be cured by a nature-based solution. And this is an area where the future of medicine perhaps lies, especially when we see the rise of antimicrobial resistance, where conventional antibiotics are failing us. And this is something that I want to share with you. Two important examples where people were saved by using phage therapy. The person I don't know whether all of you recognize this, but this is an enlargement from the Beatles album called Sergeant Pepper's Lonely Heart. Now, this is an enlargement because on the right, you would see John Lennon in the yellow jacket. The two gentlemen on the bottom left are Ringo Starr and Paul McCartney. Above Ringo Starr is Marlon Brando. But the person of interest to us in this image is the gentleman with the big hat. Now, this gentleman is goes by the name of Tom Mix. During the silent era, he was the single largest superstar of the Hollywood world, especially in the cowboy films. And there were cereals and soup cans that were named after his name. Um, but in November 1931, at the onset of the talkie cinema, there was something that happened to Tom Mix, and he fell down. He developed a very bad case of peritonitis. Thankfully, he was in Hollywood, in Los Angeles. The Stanford Medical School had a very vibrant department of microbiology. They were able to diagnose the peritonitis. They were able to identify and classify the bacteria that was causing the peritonitis. They in their stock they had a collection of vials which were targeted to this specific bacterium that was causing the peritonitis, and when they applied that in the bowels of in the gut of Tom Mix, who was there was no antibiotic in 1931, not no effective antibiotic. Penicillin was still a few years away, and sulfonamides were still in their very early days, and they were not effective on peritonitis. So when the phage were used on Tom Mix, they worked wonders, and he was up and about, and his Hollywood career resumed. The other person is somebody you might all recognize. This is Elizabeth Taylor. In uh, in May 1961, she had returned from Egypt after shooting for the film Cleopatra. But as soon as she arrived in London, a day or two later, she collapsed in a hotel, and it was found that she had a severe case of pneumonia, and they had to do an emergency tracheotomy in the hotel itself because she was unable to breathe. She was gasping for air, and she was taken to a hospital where the conventional penicillin was not working. But there was a new type of penicillin that was discovered called methyl penicillin. But at the same time, the doctors were saying that they could not take the risk of using just methyl penicillin because it wasn't proven to work against the deadly Staphylococcus aureus. They knew that there was a lab in Philadelphia, which had bacteriophages that worked very well on Staphylococcus aureus. So they called out for that, and there was a special shipment that left Philadelphia from the Delmont labs, the one I was telling you about, for that continues to make bacteriophages for cats and dogs, but not for humans. But it perhaps alongside with methyl penicillin worked and saved the life of Miss Taylor. But sadly, what happened was that the New York Times and other papers highlighted the importance of methyl penicillin and forgot to mention phage therapy. And as a result, phage therapy has since then been forgotten. Now, here's something which is non-toxic, very specific, and exits the body, the human body, as soon it finds that its host, in this case Staphylococcus aureus, as soon as it has exterminated all the Staphylococcus aureus, the virus will leave your body because it doesn't find any more Staphylococcus in your bloodstream. So something as clean and effective has been ignored for several years, and I think it's time that phage therapy comes back into practice. And my final point, and I'm sorry, organizers, that I'm taking longer, but this is going to be quick and about to end my presentation. Um, Without viruses and the microbial world, our world would be less beautiful. And let me just show you what I mean. Tulips, wild tulips, are usually single colored. Some of them are two colored, but they're small and they're not as impressive when you see them in the wild. They look like normal lilies. They're actually closely related to the lily family, but they're not something spectacular. But once the bulbs of tulips left Venice and reached Spain, and from Spain they went to Holland, and this is in the late 1500s. They encountered a virus that was found in trees, which served as a reservoir. The trees that I were of apricots, cherries, and plums, and those kind of trees. And there was a common virus that was found there. It also could infect potato when potato was introduced in Europe, and it's called the POTI virus. POTI standing for POT is potato, and Y is Y virus. So potato Y virus was the one of the viruses that infected the bulbs, and it caused the tulips to become spectacular. From being very predictable red and yellow or plain colored, they started to develop streaks and lines and different colors. In fact, at one time, the craze for tulips became so grand that ships laden with spices and linen and silks from India and China uh, could be purchased by using just a single bulb of tulip. If it was grand and beautiful, that's the price that people were willing to pay for it. In fact, the craze went so out of hand that the first economic bubble in the world is actually called tulip mania. Now, tulips actually caused such a frenzy in Amsterdam and in much of Holland that people were betting on and putting their homes and warehouses and factories and ships on mortgage just to own a very fancy tulip. And once that came crashing, the first economic bubble also burst open. My second proposition is that this is how wild grapes look like. Now, the red thing that you see is called a pedicel. Now, pedicel is a wasteful thing because it's not edible. In fact, the wild grape itself is not edible because it's very bitter, the seed is large, and the pulp is very acidic. But what the virus has done for us, there have been three successive viral infections that created the bunchy grape that we know. I'm not going to go into the details. Again, please buy my book. Uh, Rice in wild. Now, this is a picture that I've taken in Jorhat. This is how the wild rice looks like. Notice that the spikes and spikelets at the bottom and on the top have not matured, right? They are not ripe. They're not ready for harvest. But a series of viral infections have ensured that the rice that you and I consume, that has been now been agriculturally possible, is something that ripens simultaneously. And this is something that has been uh, 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 an endowment given to us by the viral kingdom. So, and my final submission is that the American chestnut, these ghostly images that you see of trees, bottom right, just for scale, you can see a car, a very tiny car. It looks like a Tempo, but it's actually a huge Ford T. But the chestnut forest of America was completely devastated by an imported fungus. And once that fungus uh, had taken root in just about every chestnut tree, the trees would die a very slow death. But then in Europe, some scientists found that there are two kinds of fungus. One is the extremely belligerent and virulent orange colored fungus, the one that caused the decimation of the American chestnuts. But they also found a benign version of it. It was a gray fungus which had a virus in it. But if you apply the exudate of the gray benign virus, chances are that the orange virulent variety turns gray and becomes absolutely benign, and the tree would survive. Now, the whole process of recovery is taking place after 50 years or 60 years of its discovery. And slowly, the chestnut forests of America are recovering thanks to a virus that lives within the fungus that actually causes that disease. So, by finding a virulent version, and its own antidote in the body of the fungus, we're now able to use it against itself. So the virus actually neutralizes a virulent fungus. And my final takeaway points are this: why did I write a book on viruses when everybody was grappling and cursing the microbial world and viruses in particular? I thought there is something more to the virus story, and I think I've tried making 5 submissions to you. I have 7 other submissions in my book. Please go ahead and read it. Sorry, it's a hard sell by our author. But the point is that nothing in nature is wasteful. We are the ones who assign or castigate that wastefulness, that this is evil and this is good. And we also label life forms as bugs and critters, slimes, weed, gunk, whatever it is. Now, I think we do this at our own peril. If we are calling blue-green bacteria or cyanobacteria as slime or gunk or whatever we want to call it, we are being unfair to ourselves, because these are the creatures that are producing all the oxygen and sinking our carbon into the depths of the oceans, right? The same thing is with the viruses. I think it's time we stop generalizing and labeling things wrongly. And it's not only good for science, but it's also good for public relations and for making a better uh, perspective, building a better perspective when it comes to the public. Because once something which is creepy and crawly is thought to be bad, everything has to be killed. That's not true, you know. So my fervent plea is that please do not label all life forms as being derogatory or being out there to get the human race. That's not true at all. The more important thing is for students of IIT Jodhpur is to question everything. They cannot be a very simple, straightforward answer to complex natural phenomena. And for this, you need to stay curious, you need to ask questions, you have to ask for counterfactuals, you have to get into critical thinking, and you need to continue to question the methodology, the findings, and the rigor with which science gives you answers. Thank you so much for this opportunity again, Dr. Shantanu Chaudhary and Professor Mitali Mukherjee, and everyone at IIT Jodhpur for inviting me. Thank you so much.